The rupture of intracranial aneurysms accounts for about 500,000 deaths alone every single year worldwide. My name is Sam Khodursky, and today I'm going to be talking about using GWAS to understand the genetics of intracranial aneurysms. So first off, for some background, specifically on epidemiology, intracranial aneurysms have a prevalence of about 3%, and about 0.3 to 1% rupture annually. As I previously mentioned, rupture accounts for about 500,000 deaths every single year worldwide. There are various risk factors, but some of the more prominent ones include hypertension, tobacco and alcohol use, and female sex, and also family history, as you can see in this nice little cartoon. Now I want to give you some background on biology and anatomy. Most of these aneurysms occur at bifurcations within the circle of Willis. A large majority are found in anterior circulation, so the branches coming off the ICA. And the overall biology of why they form is thought to be the result of an interplay between hemodynamic stress, such as in the case of hypertension, and inflammation leading to diminished vessel wall integrity. So why would someone study intracranial aneurysm genetics? Well, for one, we could use the genetics underlying IA to screen individuals and find individuals that are at risk of rupture or at risk of having intracranial aneurysms so that we can intervene in time. And second, we could identify druggable targets to help prevent intracranial aneurysms from forming or already formed intracranial aneurysms from rupturing. I want to give you some background on genetics as well. So one concept that I want to talk about is liability. And every single person has a liability for every single binary phenotype or disease. And you can think of it as a number. And when this number crosses a threshold value, you get the disease. And a liability is the sum of genetic factors, environmental factors, and noise. Another concept that I want to talk about is heritability. And that's a term that gets thrown around a lot, but for binary traits, it has a very specific definition. And that is the proportion of variance in liability that's explained by genetics. And by variance, I mean variance between individuals. For intracranial aneurysm specifically, the heritability has been estimated to be 41% from twin studies, and the majority of that heritability can be explained by common variants. And I'll talk a little bit more about the GWAS that produced those results a bit down the line. Before I get into GWAS, I want to briefly mention monogenic disorders and how they associate with intracranial aneurysms. So autosomal dominant polycystic kidney disease has shown to be associated with intracranial aneurysms. Other connective tissue monogenic diseases such as Ehlers-Danlos, Marfan, and Loeys-Dietz syndrome have also been shown to be associated with intracranial aneurysms. And additionally, microcephalic osteodysplastic primordial dwarfism has also been shown to be associated with intracranial aneurysms. But importantly, monogenic disorders account for only a small fraction of intracranial aneurysm patients overall. So now I want to talk a little bit about GWAS. If you remember, a couple slides ago, I mentioned that a majority of the heritability in intracranial aneurysms can be explained by common variants. So what is GWAS for? Generally, when we think of Mendelian traits, we think about rare, large effect variants. But GWAS, or genome-wide association studies, allow us to identify associations between common variants and various phenotypes or disease. So how does GWAS work? Using this example from Wikipedia, you can think of GWAS as a test that tests every single variant in a genome. And at every single variant, the scatter plot shows one specific variant. You look at the different genotypes And as you can see in this example, the CC genotype has a lower average phenotype than the TT genotype. And what you do is you fit a linear regression and you extract the slope and you test for the significance of the slope. And that's the p-value at that site for a GWAS. And the slope would be the effect size. When we're thinking about binary traits, such as intracranial aneurysms, we use a logistic regression instead of a linear regression. At the end of the day, a very common way to plot the results is to use something called a Manhattan plot, as you can see on the bottom. And on the y-axis of a Manhattan plot, you have negative log 10 p-values, so the higher the point, the more significant. And on the x-axis, you have variants as they're positioned along the genome. And by the way, this is a GWAS of, I think, kidney stones that I got from Wikipedia. Finally, I want to get to some real data for intracranial aneurysms. Here's a Miami plot. So a Miami plot is like a Manhattan plot, but there's a Manhattan plot on the top and a Manhattan plot on the bottom. And this is from a study by Bakar et al. And it's a meta-analysis of GWASs for intracranial aneurysms. The top Manhattan plot shows a meta-analysis of European individuals, and the bottom Manhattan plot shows a meta-analysis of European and East Asian individuals. So the bottom analysis actually has a bit more power, and they identified 17 independent loci that were associated with intracranial aneurysms. More recently, a study came out from Adkar et al., which identified 5 additional loci that were associated with intracranial aneurysms. Both the Bakar et al. And Adkar et al. Studies found that the most significant association occurred at a locus that codes for a long non-coding RNA called ANRIL, which is associated with several cardiovascular and neoplastic phenotypes. Other genes that have been prioritized from the Adkar et al. Study include SOC17 and FGD6. Both are involved in endothelial cell signaling. BCAR1, which encodes a mechanical stress sensor, and TWIS1, which encodes a transcription factor involved in the regulation of smooth muscle cells. Out of all of these prioritized candidate genes, one has actually been functionally validated, and this was actually functionally validated before the GWASs took place. And I'm specifically talking about SOC17. As I briefly mentioned, it's expressed in endothelial cells. A study from 2015 by Li et al. Basically generated mice that have deficiency in SOC17, and they simulated hypertension in these mice by infusing angiotensin II. And this resulted in intracranial aneurysm-like vascular abnormalities in the mice. As you can see in this data from that paper, if you look at the I delta EC, which is just the SOC17 deficiency, and the I delta EC plus AT2, the angiotensin II infusion, you can see that these aneurysms have a larger luminal diameter and have decreased wall thickness. Additional data in that paper showed that SOC17 deficiency causes aneurysm formation through impaired PDGF beta secretion. So what else can we learn from the GWAS data? Well, Adkar et al., they prioritized the list of genes by using GWAS in combination with other statistical approaches, and they analyzed some single nucleus RNA sequencing data from humans, and these individuals did not have known intracranial aneurysms. And by looking to see where their candidate genes were expressed, it could give us a sense of which cell types are important for intracranial aneurysm formation. And what they found is that smooth muscle cells, pericytes, endothelial cells, and fibroblasts likely contribute to intracranial aneurysm development. And finally, I want to talk about polygenic risk. So what's a polygenic risk score? It's a quantitative metric of an individual's genetic susceptibility to disease. And it's calculated as a weighted sum of thousands of variants associated with that disease. And the weights used for this weighted sum come from GWAS. And overall, polygenic risk scores are used to estimate the likelihood that a person will develop that phenotype or disease. And one can think of this as an attempt to calculate genetic liability, as a throwback to one of the earlier slides. So using the 2020 Nature Genetics GWAS data from Bakar et al., Adkar et al. Constructed a polygenic risk score using over 700,000 variants. And what they found was a high polygenic risk score was significantly associated with increased risk of intracranial aneurysm. However, it was only about as predictive as simple discriminators such as female sex and smoking status. So there is some sort of predictive ability, but it's not particularly impressive at the moment. So now I just want to talk about conclusions. One big takeaway is that intracranial aneurysms are a polygenic phenotype. Indeed, 22 independent loci have been shown to have significant associations with intracranial aneurysms. SOC17 has been functionally validated, but additional work needs to be done to validate other genes and to see which genes can be targeted with drugs to ensure that we can develop therapies for the prevention of intracranial aneurysms. We learned that expression patterns of candidate genes from GWAS studies implicated smooth muscle cells, endothelial cells, pericytes, and fibroblasts. And finally, we talked a little bit about polygenic risk scores, and it seems like polygenic risk scores for intracranial aneurysms show some predictive ability, but further work needs to be done to make them more useful. In particular, GWASs will likely get larger over time, and GWASs will include rarer and rarer variants. So hopefully over time, these polygenic risk scores, in combination with known risk factors, can give us a real sense of who might be at risk for developing intracranial aneurysms. Thank you. That's all I have for you. And here are the references.